As I told the last group, I got into public plant breeding because I wanted to get rich, which is a joke. Yeah, so really what happened was that there was a lot, so Carl really pioneered a lot of the private licensing in the East, was one of the early adopters of it and released quite a few varieties under private license. And that was how he generated a lot of the royalties that could come back and then, they come directly back to the program that are used to fund the research and the varietal development that happens. The issue was that we're so busy out here making new varieties, we don't have time to get out there, just chatting with boots on the ground, talking to farmers. It's, this exhausts all of our time here. So what we want to do is start to diversify where our income comes from, without sacrificing the varietal development, which is a little bit of a challenge because every time you, so for example, I would say that our program was 90 to 95% varietal production and 5% science. And in particular, I am interested in doing more science, but layering it on top of the breeding. So I want to do 90% varietal production and 50% breeding technology, right? Working on science, new breeding methods. So that, for those of you that are familiar with math, 90 and 50 don't add up to 100, which means I really am trying to layer, some of the science that we're doing on top of our breeding efforts. So that includes things like this trial. So this is part of a $1.6 million grant funded by the Foundation for Food and Agriculture. This is something that I put together with my previous advisor, Kelly Robbins at Cornell, kind of as I was negotiating my terms for this position, which was nice because then, finally it gets funded and I've got, money to do some research coming in. Turns out it's pretty hard to hire people in the middle of the pandemic, so we're still looking for a postdoc right now that's got the skills we need. But I think we found one. The problem is he's in Brazil. But and Brazil's no travel right now, right? No travel ban, so we're trying to figure that out at the moment. But anyway, so what we're trying to do is layer some of these breeding technologies. What do I mean by breeding technology? I mean incorporating high throughput phenotyping, doing things like aerial imaging, incorporating genomic information, and all of this is to drive breeding decisions, right? Both decisions surrounding advancement. Okay, I have this one, should it go to next year's trials? As well as decisions surrounding, should I include these individuals as parents? Okay, especially earlier in the program, it's nice to be able to start to include early material as parents, because then you're reducing your cycle time, and that's how you really accelerate gains, is by reducing cycle time. So one of the things that we wanted to do is look at, so for those of you that saw Wade's talk, I think that it's pretty clear that it's getting warmer. But I don't know if you saw that graph or not, but it has this very nice linear trend, but you'll notice in the early years it kind of does this, and then in later years it starts to do this. Okay, so it gets that shape gets wider, right? So we've got a lot more variability, and what variability in environmental variation causes is differential genotypic response. Okay, so we call this genotype by environment interaction, and that's effectively the idea that some varieties will do very well under some environmental conditions, and other varieties will do well under different environmental conditions, and that there is not a single variety that does the best in all environments. And that's a old plant breeding problem that we've known about for a very long time. Most of what has been done in that area has been what we call multi-environment trials, where we plant the same lines in a bunch of different environments, and then kind of retrospectively we can look back and we can say, okay, well, we can build a story about why this one did well in that environment or under those conditions, and why this other, or why it failed here, and why this other one, you know, didn't do well here but did very well over there, right? So we do a good job of that, but it's very black box. We don't really understand a lot of why. Okay, it's kind of, we come in at the end, and you know, we come in the end. We say, okay, well, I know it heads a little earlier, and I'm going to combine it. I'm going to get the, you know, the plot yield and the test weight and the moisture, and kind of using some of that information, you can build this story. We haven't been able to measure longitudinal traits. And what I mean by longitudinal is traits that you can observe through time, like growth. Okay, because the only way to do it is to tell your graduate student that he's got to come in and he's got to go take plant heights on every single plot every day from now until, you know, or from November until, you know, July. And that's obviously a very cruel thing to do, so we haven't done that, which means that we largely have not observed traits as they change through time. But now there's stuff out there that allows us to start to look at longitudinal traits like growth and development. And those are some of the things that we're starting to ask. How can we now relate those growth and developmental changes directly to environmental conditions that we've observed and environmental conditions that we expect in the future? All right, and that's really the key is some of this, what is going to happen tomorrow, right? So out here we have a couple of drones. One of them lives here in Warsaw. The taller one over there lives here in Warsaw. The shorter one lives in Blacksburg. They're both equipped with multispectral cameras that kind of sample the visual spectrum as well as the far red and the near infrared. And using linear and nonlinear combinations of those bands, we can get estimates of, say, plot photosynthetic capacity. We usually do that through normalized vegetative difference, which is an index, NDVI. Of course, there's all kinds of other indices that we could use with these various, you know, wavelengths, these light bands that we're getting, reflectance. But so for right now, we're mostly just focusing on NDVI. It gives you a kind of a instantaneous estimate of photosynthetic capacity. Okay, and so then we can kind of plot that through time, right? And then you can fit a genotype, a growth curve for every genotype to those, right? And this is all done using genomic information. We allow sharing amongst sharing of genetic information amongst individuals using random regression models. Anyway, that stuff's not that important. But we can build these growth curves, and then we can kind of integrate across that growth curve to look at total photosynthesis over time, as it builds up over time. And so then what we want to know is what is it about the shape of those curves that leads things to be either very sensitive to environmental conditions, where they do particularly good in this environment, but, you know, really poorly in that one, or relatively stable, right? So in plant breeding, we often want to maximize sensitivity to predictable differences. Predictable meaning like location, soil type, that kind of thing. We want to minimize sensitivity to unpredictable variation. That's like year-to-year variation. So, you know, temperature, rainfall, those kinds of things. When we build these genotypes, so we come in here, we image these things, say, every week, try to capture especially important times like heading and maturation are very important, because we want to know, we want to get those time points where there's really big phenotypic differences between these things that at least we can see with our eyes. And so we come in here, we build these curves, and then we ask the questions that are associated with, okay, so is it better to have deterministic growth, where it doesn't really matter what the environment is, you just march along, right? And if it gets hot or dry, well, then you just keep cruising, and then you run out of water, you keep transpiring, you keep photosynthesizing, and then eventually you die, right? Or is it better to have strategies where you're actually regulating more and responding to the environment more, being a little bit more plastic in your growth and development, and allowing you to say, oh, well, it's really dry like it is now. Maybe I should slow down that transpiration kind of reserve some of that water in the soil now, so that I can hold out through this entire period until we get rain again. Okay. Now here we're working in wheat. When I say that we're kind of layering these breeding goals, we have. Oh, thanks, buddy. These mayflies are a little annoying. Our apologies. They're almost done. I think they were pretty bad earlier in the week, but they only last a short while. So yeah, I was talking about layering. So here what I've done is I've put a lot of our breeding materials in here so that we get information from stuff that's actually moving through the program, right? These aren't just lines we kind of chose at random. We chose there's a couple of bidirectional selection experiments in there. There's some historical material, but I've got a lot of my first year selections in there. I've got stuff that's in the state test in there. So we kind of want to, start looking at active breeding material all the time. How can we connect the information that we get from these high throughput phenotypes to making those breeding decisions? Okay. So I've been talking mostly about wheat, but this is a much larger project. There's alfalfa at New Mexico State, another alfalfa experiment at Cornell University with my former PI. Actually, the one at New Mexico State is also my master's PI. Partners with BASF and Lima Grain, they're looking at corn, cotton, canola. So we're kind of sampling a large breadth of different geographies through time. So it's a 3-year project, as well as different species, and we're kind of looking, what are some of these strategies? Can we start to use this type of information on these longitudinal traits to make informed breeding decisions? And so this study is kind of sampling that environmental variation just through a couple of different locations and across time. But my graduate student Cameron is working on a similar project where he actually has a explicitly designed environmental gradient. And I'll let him just give you just a few sentences on that project. You want to grab your?
All right, so kind of similar to what we're doing with this project, where we're using the drones and we're gathering images or data over time. I'm also doing that, but I'm also adding another variable of nitrogen. So we've got different levels of nitrogen, and we're seeing how individual lines or varieties respond to that. And another goal of my study is to use this data and also use that data that we've collected with the drone along with genetic information, to find regions of the genome that are associated with high nitrogen use efficiency or low nitrogen use efficiency. And so we have that as a goal. And we're going to use that information to look into how we can breed for wheat that uses nitrogen more efficiently.
And with that, I think we'll take some questions, and then we'll let you get on to the next station. I don't remember. Do you guys have one more? I think so. This is your last. So I'm standing between you and lunch. If you're ready, please feel free to head back. Also, feel free to check out the study. I would say there's a very interesting little piece in here. Part of this is a ground truthing experiment where we had the same 22 lines that we replicated many times. And throughout the growing season, we've come in and we've cut them down to the ground, collected all that biomass, and then weighed it. We did some forage quality on it, that kind of a thing, just to see what's going on. And it's mostly to get a ground truth for the drone, right? What is the drone actually seeing from above? Can we get like real one-to-one connections with what we have on the ground? And is that genetic, or is it just phenotypic, meaning is it just environmental? And so down there, the fascinating thing about it, and I encourage you to go down there, we cut a whole rep of those, so it's 2 ranges down to the ground at about growth stage 30, 31, something like that, and we cut them completely down to the ground. We did all of the stuff with the biomass we wanted to do. I come back and they are in the middle of flowering again. And I am just fascinated to know how much of that, right? So I cut off, you got that growth curve, right? And you're integrating across it, building up all that photosynthate, okay? I cut off the tail of it and I threw it away. And then it had to start from scratch again. do it again, and I'm shocked how many heads are over there. I can't believe that they were able to recover. It's really wild.
Well,
so
see,
it's
you'll
kinda
get
neat.
people that talk about that who are in the western part of the state. Some of them are talking about in other places west where they want to know how long they can graze it with livestock
Right.
and then pull them off
And still get the
then
crop.
still get the crop.
Yeah.
And with awnless varieties, that may be as important as anything because they're talking about using it for forage later on. So if they can still graze it and then cut a forage crop of it, they might not ever put a combine to it, but it's another use for that wheat crop or small grain crop. It's not traditional you see out here in the east.
Right. Yeah, it's cool stuff. Yeah, definitely. And you know, kind of rethinking how we might use good products that we have today for different purposes is super important. Absolutely. Great. Well, I don't want to keep you here. If you're ready.